into much smaller molecules, which then are transported to the circulatory system, and those can be carried to the cells of the body, and the cells can use these fatty acids and amino acids and sugars for fuel. So anyways, this figure, the information on this figure will be on test number 1. I'm going to stress the things I want you to know about this figure. And I'll try to keep it relatively simple. And that is out there under course contents. And I think that's it. Yeah?
When we're doing the lab write-up, do you want us to use the conglomerated data or individual data that we got?
You're going to use the data that's actually posted out there. So you'll see a file that has the data that I got from those of you who emailed it to me.
Okay.
So that's the data you're going to use. So here's a very simplified version of what I presented to you thus far. We talked about the breakdown of glucose into pyruvate. And then when oxygen is present, pyruvate is shuttled into the mitochondria, and we're able to further break that down, and we end up making a fair amount of ATP. There's a lot of bookkeeping, and you need to know all the numbers that I put up there on the board. I wouldn't put them up there if they weren't something you need to know. I finished lecture Monday night with a summary equation for what we refer to as aerobic glycolysis. Aerobic glycolysis is the breakdown of glucose into pyruvate, and then the pyruvate being shuttled into the mitochondria, and then from this we capture all these electrons, we transfer energy from these high energy electrons, we make a proton gradient out of the energy we've taken away from the electrons. There's one of our fundamental principles, one of the 8 fundamental principles, which basically says that there's energy stored in ion concentration gradients. And we build this huge proton gradient, and we use that huge proton gradient to actually drive a proton pump backwards, thereby making ATP. And so when glucose is the substrate or the fuel for this process, from a molecule of glucose, we end up making 36 molecules of ATP. Now, as you're sitting there listening to me right now, about 60 to 70% of the ATP that you're making is coming from fatty acids, not from glucose. If I were to take you to the gym and we all went and worked out on a treadmill, as we increase the speed on the treadmill or the speed at which we're walking or running, then you shift from a predominant use of fatty acids to greater and greater glucose utilization as the intensity of exercise goes up. And you can vary the fuel utilization of glucose and fatty acids depending upon your metabolic state. And what I mean by that is how active are you. But right now, as you're sitting there, about 60-70% of all your ATP is being made using fatty acids. Well, here I'm just going to write glucose. How about when we're using fatty acids? And here's this formula here is meant to represent palmitic acid. And so you can see the formula is a lot different. Now, just a quick timeout. I expect all of you to have this chemical formula on top memorized. The one on the bottom, no. There's a point I'm gonna make. The point I'm gonna make, I expect you to understand and to be able to properly address any issues I raise with regards to that. So here's palmitic acid, 16 carbons, 23 hydrogen, and 2 oxygen atoms. In this reaction, we combine that with 23 molecules of oxygen, and we take 129 molecules of ADP, add 129 molecules of phosphate to that, we make 129 molecules of ATP. You can see that over here. We use 23 molecules of oxygen, we produce 16 molecules of CO2. This ratio between CO2 production and oxygen consumption is important, and that's what I want you to know. So there's this term that's referred to as the respiratory quotient. And this is abbreviated as RQ. Isabel, you ever hear the respiratory quotient? You have? What is respiratory quotient? Can you tell me?
I think it's if you're evaluating how much CO2 is being breathed out, you can determine what's happening metabolically on whether or not you're using more fatty acids versus carbohydrates.
Okay, good. That's good. That's a good shot at it. So the respiratory quotient, RQ, is equal to the rate of CO2 production. I'm going to put capital V, and I'm going to put CO2. In biology land, in chemistry land, a capital V would represent volume. However, we're in physiology land. And what I'm going to do is I'm going to put a dot above the V. And anytime you see a symbol, I don't care what it is, with a dot above it, that means volume per unit of time. That's a rate. So here I'm going to put a dot above this V CO2, and we're going to read that as V dot CO2. And that's the rate of CO2 production. Divided by what's referred to as V dot O2. And that's the rate of oxygen consumption. Well, how do we get this? And the answer is here. If I were to just look at the board. Here's the rate of oxygen consumption, 602. And here's the rate of CO2 production. Where's Mr. Cortez? Chris Cortez. Chris, if glucose is the only molecule that's being used to produce ATP, then you're gonna tell us that the respiratory quotient will be what?
I'm not sure.
Okay, anybody? Nobody? Yes, go for it.
Is it one?
It will be one, that's exactly right, because when we're talking about glucose, it's simply six CO2 over six O2, and so you would sit there and say 6 divided by 6 is numeral uno, 1. So what's cool is that we can have somebody sitting, resting, exercising, and we can put a mask on them. And as they're working or just sitting there, we're actually measuring the rate at which air flows into and out of the lungs. We're measuring what's happening to the oxygen coming in the lungs, what's happening to the oxygen leaving the lungs, the same thing for CO2. And we can actually measure the production of CO2 and the consumption of oxygen. And so we can tell fairly accurately what fuel source the individual's using. Well, that's if we're talking about glucose. So whenever we're talking about glucose as our fuel, you're going to tell me that the respiratory quotient is going to be 1. How do we know that? Because here's the summary equation which tells us about the ratio of CO2 consumption, oxygen consumption to CO2 production. Let me just stop there, ask if there are any questions. Don't be bashful, speak up. Come on, yeah, go for it.
Can you repeat again why there's a dot on the top V and at the bottom V?
Yeah, the dot is a convention that's used by scientists to indicate that we're talking about something that is a function of time. It's a rate. So if I have a V with a dot above it, that means volume per unit of time. That per unit of time could be a second, it could be a minute, whatever time units you want to use. But typically in physiology land, we would be talking about liters of CO2 produced per minute or liters of oxygen consumed per minute. Did that help?
Yeah, thank you.
I'm pretty sure it is W dot, you just made it less emphasized.
Oh, sorry.
All right, well, what if we're not using a carbohydrate or using glucose, but we're using a fatty acid? Well, here's the equation, the summary equation for the use of palmitic acid. And in this instance, when we sit there and we take a look at the oxygen consumed and the CO2 that's produced, we see something that's different. We see that the ratio is 16 CO2 produced versus 23 molecules of oxygen that are consumed. Okay, break out your calculator real quick and tell me what's the respiratory quotient. 0.6956. I love the way she said that. Can I round that off to 0.7? Good. So when we're using fatty acids exclusively to make ATP, the respiratory quotient is going to be 0.7. This next part you have to listen to very carefully. Students don't get it initially, but it's easy. When glucose is responsible for 100% of the production of ATP, when glucose is responsible for 100% of the production of ATP, the fats are responsible for how much?
0.
0. That's logic 101. when glucose is responsible for all the production of ATP, the respiratory quotient is 1. When fatty acids are responsible for all the production of ATP, the respiratory quotient is 0.7. By definition, at 0.7, glucose accounts for how much ATP produced? 0. So our range goes from 1.0 to 0.7. Yes.
What happens if you have like 0.85?
Give me a moment. Can I go ahead and raise this? Everybody good with this?
You said we need to memorize the glucose one?
That's correct. You need to know
That's
that.
what Paul meant, yes.
Yeah, just the one for glucose. So I'm going to put a column here, and I'm going to put ATP from glucose. I'm going to put another column, ATP. I'm going to put percent, just so it's clear. Percent ATP from fatty acids, FA for fatty acid. And I'm going to put over here in the third column, respiratory quotient. And we've already gone through this, but just repeat what I've stated. When the percent ATP from glucose is 100%, by definition, the percent from fatty acids is 0. And the respiratory quotient will be 1.0. How do you know that? Because the summary chemical equations I had up there tell you the relationship between oxygen consumed and CO2 produced. Well, if the percent of ATP produced from fatty acids is 100%, by definition, none of the ATP is produced from the use of glucose. And we know that the respiratory quotient under these conditions will be 0.7. So simply stated, as we go from using only glucose and transitioning to using fatty acids, we go from an RQ of 1 down towards 0.7. Now, Isabel just sat there and said, well, what if the respiratory quotient is 0. Did you say 8.5 or 8.3?
85.
8.5. Well, the delta between these two extremes, these are the extremes. You can't go any higher above 1, and you can't go any lower than 0.7. That's what I'm telling you. In the real world, yes, you can go above 1, but I'm not going to get into why that is. So the delta, if these are the two extreme conditions, then the delta between those two, hopefully you're all going to sit there and tell me that it is 0.3. Well, hopefully all of you would tell me that 1/2 of 0.3 0.3 is 0.15. And if I add 0.15 onto 0.7, I get 0.85. 0.85 is, R cubed 0.85 is right in the middle of these two extremes. So we've got 0.85. Where's me? Me. Based on what we have up there, you would predict that the percent of ATP that is coming from glucose is what? If the R cubed is 0.85, how much of the ATP, what percent of it is coming from the use of glucose? It is 50. And what percent of the ATP is being produced from fatty acids? Well, the respiratory quotient is 0.85. It is 50, that's exactly it. We're going to say that right now, as you're sitting there, that you have a respiratory quotient of about 0.8. Why do I use that? I use that because that's a pretty close real number for people who are in a basal state, a resting state. So what I want each of you to do is break out a calculator, and the hypothetical is that you measured your respiratory quotient, you found out it's 0.8, and the question is, how much of the ATP is coming from the use of glucose? How much of it is coming from the use of fatty acids? Go for it.
Or which one? FA? No, glucose. Wait. Glucose 45. FA 55. Why don't let's do that. I think you pick the and then it's 3 to 0.1. I think it's 60-40. I'm guessing. I'm guessing it's 45-55. There's no reason. I'm guessing nothing. Just as if we're going up by I get where your cell process is coming from though. 85. I feel like it's gonna be so complicated number like 47. So that's the 5 minutes. I'm not the color. Do I ask him how can we input in a calculator?
Mr. Mead, or Scott?
Yeah.
So Scott, if the respiratory quotient is 0.8, that's what you measure. Then what percent, well, let me back up. If you're at 0.8, are you closer to producing most of the ATP from a fatty acid, or closer to the RQ associated with mostly glucose?
Fatty acid.
Fatty acids. So it's got to be greater than 50%. We're basically biased so that the percent fat is going to be higher than 50%. How do we know that? Because R cubed 0.85 is 50-50. Our absolute delta is 0.3. At 0.8, the delta is 0.2. So we've got 0.2 divided by 0.3, and your answer is
77%.
66%,
66%, yes, sir.
right?
Yeah.
Yeah, well, maybe so. The delta here is 0.3, going from 1 to 0.7. We're biased towards a greater use of fatty acids than we are glucose. And from 1.0 to 0.8, that delta is 0.2. And so all you have to do is take the delta that you see here, and in this case, the RQ equals 0.8, and the delta for that, in terms of our substrate utilization, is 0.2 divided by 0.3. So by definition, we know that 66% of the ATP is coming from the use of fatty acid. And of course, that means that glucose accounts for what percent of the ATP being produced?
34.
And the answer is, not the
34.
remainder. You could say that, but you wouldn't get any points for that. You'd sit there and say 33%. So all of you should be able to calculate the percent of ATP coming from glucose and fatty acid by knowing the respiratory quotient. And if we're to have each and every one of you do an exercise test, you'd start out 0.8, and as we increase the intensity of the exercise, you keep creeping up higher and higher and higher until eventually you got at 1.0. Glucose is the preferred fuel during physical activity. Yes, ma'am?
So we did how to get the percentages if you're given the RQ. How could you figure out the RQ if you were given different percentages? You were given like 25, 75%.
So 25, 75%? I'm not quite following you there, because if I had 25%, 25% of what?
Glucose, 75%. Okay. So if you
Then-
were given the percentages, how would you figure out the RQ from being provided the percentages?
Okay, so if we had 25% glucose, then basically what we're gonna do is, you wanna know the respiratory quotient associated with that?
1%.
Yeah, so you would sit there, here's your maximum delta, you take 25% of that. Right, so 25% of 0.3 would be 0.75 or 0.075, something like that. Anyways, it's easy to do. All right. So that's known as the respiratory quotient. I also want to introduce this term to you. And this term is called a MET.
Am I here?
M-E-T, MET. Yes.
When we were talking about if we put the RQ to 0.8 and then we came out with 66%, you said the remaining number is 33, wouldn't it be 34? Does it matter?
Well, if we did the math, it'd be 66.66666,
Okay.
right? And then it'd be 33.333333,
Okay.
right?
Got it.
So we're just looking to be in the ballpark.
Okay.
If you sat there and said it's 20%, that's wrong. If you sit there and say 66%, that's right. If you sat there and said, oh, glucose would be 33.3333%, fine, you got the answer.
Okay.
Yeah. Isabel, do you guys use the term MET in nutrition?
I don't.
Does the field use it?
I think so.
A MET. A MET is a metabolic equivalent. And it is basically the metabolic rate divided by the basal metabolic rate. So what do we mean by basal metabolic rate? If you could imagine, right before you go to sleep, you're inactive, you're lying down, hopefully, and you're in the least metabolic state, the lowest metabolic state you can be in. That's the basal metabolic state. So what we'd like to know is, what is the relative metabolic level of an individual? If you go to 24-hour fitness or Orange Theory, or you can afford to go to LA Fitness, or what's a really high-end exercise joint these days?
Renaissance.
Equinox, there you go. Sorry, I go to the cheapest place I can find, 24-hour fitness. No joke, I went in a month ago to log in after I've been going there for 20 years. And it says, talk to the attendant. Here's what they did to me. I was paying $7 a month because I had become a member 20 some odd years ago. And what they did is they changed their membership rules, so they have different tiers now. So I went from $7 a month, and it was really hard for me to accept this, but I went to $21 a month. Anyways, having a little fun with 24-hour fitness. Anyways, so you have 24-hour fitness, and you're at any of these joints, you look at the exercise machines, and you may see a little button on there that might say RPMs, and you may see that says MET, M-E-T. What does that stand for? Metabolic equivalent. And what it's telling you is that if you're at a MET level of 10, that means you're exercising at an intensity that's 10 times greater than your basal metabolic rate. So all it is the relative index of the intensity of exercise based upon or using the frame of reference as your basal metabolic rate. So you might say, well, wait a minute, you got two metabolic rates here. Yeah, but one is under absolute resting conditions. The metabolic rate you're in right now could be darn close to it, or in fact, we could have you guys do some type of activity, depending upon the intensity, your exercise level gets greater and greater, your need for oxygen gets greater and greater, and therefore your MET level goes up. Any questions about that? How many of you go to any of these exercise places on a regular basis? Come on, raise your hands, don't be bashful. Got 1, 2, 3, 4, 5, 6, 7. Yeah. Questions about that? Confusion? Don't be bashful, speak up. Yes?
What did you say it was the relative index?
It's a relative index that describes how high your metabolism is relative to your basal metabolic rate, the lowest metabolic rate you can achieve. And a good example of when you're in that state is hopefully at night when you're getting ready to go to sleep. That's about the lowest metabolic rate you can have during the day.
Okay, thank you.
Yeah, and there's a cool circadian rhythm where people go to sleep and their metabolic rate decreases. And then something very fascinating, once you get around about 4 a.m. to 5 a.m. in the morning, your circadian rhythm starts to shift gears and you start to increase your metabolism. Even though you're not aware of it, you begin to increase your metabolic rate. And then of course, as you sit there and you start waking up, your metabolic rate increases even more. And this is one of the reasons why. Do we have anybody in here who's an EMT or has been an EMT? Anybody? Nobody? The majority of heart attacks occur early in the morning. Why is that? It's because of this change in our metabolism for going from a low energetic state as we're going to sleep to now when we start to wake up, our metabolism starts to ramp up. Really interesting phenomenon. Okay, now let's come back over here. So we've really only talked about glucose. We're starting to talk about fatty acids. And so we can talk about different types of lipids, and under the class of lipids, there are so-called fatty acids. And these fatty acids will be referred to, one group of them is referred to as monoacyl glycerols. And these monoacyl glycerols are simply a molecule of glycerol that has only one fatty acid associated with it. So here I'm just going to put Fa for fatty acid. There is another category known as diacylglycerols. And here, we've got a molecule of glycerol and it has two fatty acids bound to it. And then you can have a triglycerol, triacylglycerol. Now I'm just going to do this over here. And hopefully all of you would guess, seeing that prefix of tri, you'd sit there and say that attached to this molecule of glycerol are three fatty acids. And I'm representing these little squiggly lines as fatty acids, and I'm showing different colors because fatty acids can vary from one another. So that's our triacylglycerol, sometimes referred to as triglycerides. So underneath triacylglycerol, I'm just going to put triglyceride. So here's a triglyceride. And it comes in this cell, and it's broken up into glycerol, bless you. And fatty acids. Here I'm just going to use this little squiggly line. I'm going to put Fa above it for fatty acid. So what's happening with the glycerol? The glycerol is fed into glycolysis. The fatty acid is fed into the Krebs cycle, and there's a process known as beta oxidation that's associated with this. So here I'm just going to put beta oxidation. All right, so that takes care of triglycerides, and then also proteins are digested, and then they're broken down into the building blocks, and we talk about amino acids. And so amino acids are also used as a substrate to make ATP. And I'm not going to show it here, but just understand that the amino acids, to get across the cell membrane, they need a transporter as well. So they depend upon a type of facilitated diffusion to get across the cell membrane. Make sure you understand that these glutes, those are examples of facilitated diffusion. And what I'm saying is that to get amino acids across the cell membrane, we're also dependent upon facilitated diffusion. The amino acids are primarily fed into glycolysis, pyruvate, and although they can enter into the Krebs cycle, that's a pretty uncommon way for them to be used as an energy source. Now, a long, long time ago, in a very bad, bad world, there was a thing called World War II. If you don't know your history, go learn about it, because there are these people in our world who are very bad people. These dictators, we'll call them, like Adolf Hitler. Anybody hear of Adolf Hitler? Yeah, terrible individual. And the same thing was true over in Italy, where my ancestors came from. We had Mussolini, and in Japan. And here's what I challenge you to do, is go look at the World War II images you can find out on Google, and type in prisoner of war, and then talk about starvation, and see what you come up with. Hopefully it will shock you if you haven't seen these pictures already. And hopefully you'll sit there and be like me. Some are saying that people like that should never, ever be allowed to basically control countries and large populations of people. Now, where am I headed with this? Is that if you're food restricted or undergo caloric restriction, first of all, the body has very little glucose stored in it. So over the course of a day, for instance, we're going to exhaust a lot of the glucose that's stored in the body. Some of it's stored in the liver. And tonight, when you go to sleep, your brain is still active. It's dependent upon glucose. That's its primary fuel that the brain prefers, is glucose. And what's going to happen is that over the course of the night, you're going to use about 50%, if not more, of the glucose that's stored in your liver to satisfy the metabolic needs of the brain. So my point is, there are very few calories stored in the body in the form of glucose. There's a very rich amount of energy stored in our body in the forms of subcutaneous fat. And just using myself as an example, let's say I weigh 200 pounds, and let's say I'm at 20% body fat. That means that I have about 40 pounds of body fat. How many calories would I get out of a pound of fat?
3,500?
Approximately, yeah, that's a good number. 3,600, 3,500, that's a good number. So let's just make it simple. Let's say I've got 40 pounds of fat, and let's say that there's only 3,000 calories per pound of fat. Well, 40 times 3,000, that's 120,000 calories. Isabel, what's the daily caloric expenditure of a 200-pound individual?
Like close to 3,000?
How many?
3,000?
2,000 you said?
2,000?
Approximately. That's a basal caloric expenditure. Here's a rule for all of you to write down. I'm not going to test you on this, but here's an easy number for you. You can take your weight and multiply it by 10. That's a good guesstimate of the number of calories you need to support your basal metabolic rate. The problem is we're only at our basal metabolic rate for maybe 6-8 hours a day. That's when we're sleeping. But that's a number for you. If you're sitting going, geez, I wonder how many calories I burn up a day, just take your body weight, multiply by 10, you're there. So using myself as an example, I would be burning up about 2,000 calories a day. Imagine you're one of these prisoners of war during World War II, and you're not getting fed anything. Well, 2,000 goes into 120,000 about 60 times. So maybe I've got 60 days of calories stored in the form of subcutaneous fat. What do I do when I get to day 60? This is about, what do we do? I've used up all my glucose. I've used up my body fat stores. Where am I headed next?
It's not looking too good.
Yeah, it's not looking too good, but I'm asking, where am I going to get those calories from? Because my life's not going to end on day 60. I'm going to keep going for a while. Anybody? Yes?
Your muscles?
From your muscles, that's exactly right, because the muscles are this huge source of proteins. Your muscles make up about 40% of your muscle mass. So now what happens is I'm in dire straits, and the only place I can go now are to my muscles. So I start breaking down the proteins into what? Amino acids. And here come my amino acids to the rescue. But the problem is I'm doing this, I'm losing muscle mass. And when you go take a look at these pictures, you're going to tell me that many of these prisoners of war, who, by the way, are probably 99% men, they look like what?
Skeleton.
They do look like skeletons, sad to say. And I think I told you I was at 24-hour fitness a couple weeks ago, and there's a girl in there, I'm not joking. I mean, she's seriously close to what I call the danger zone. But anyway, so those pictures are horrific, but all of you should look at them to see what madmen do to us. We should never allow that to occur. Now, sadly, and probably many of you in here know that, and I had a very good friend that this happened to just this past year. Had a mole up here. He had not a basal cell, but a squamous cell carcinoma up here. Didn't do anything about it. Didn't do anything about it for a long time. And sadly, he paid with that for his life. But they put him on chemotherapy. Here's the problem with chemotherapy, is that a lot of people lose their
Here.
okay. Hair's not up here. But that's okay. You're right, you got me on that one. They lose their Miss Nutritionist.
Appetite.
Bingo! Yeah, all right. They lose their appetite because the chemotherapy they're receiving make them nauseous. They don't want to eat anything. These folks are analogous to what's happening to these poor people who are starved to death, is that they themselves start starving themselves to death. And so they begin to lose body mass. And the loss of body mass is known as, starts with a C, ends with an A. Anybody? Nobody. Cachexia. Cachexia. Cachexia is just a term for loss in body mass. you got any questions, I'll be bashful to speak up. Yeah, so this individual I'm telling you about went from 220 pounds, I'd say a healthy 220 pounds, to 120 pounds. And shortly thereafter, he ended up losing his life. I'm predicting, by the way, the war on cancer has been going on since when? Gotta teach you guys some history in here. That's what happens when you're with an old fart. 1974, Richard Nixon, President of the United States, came out and said that we're gonna have a war on cancer. Here we are, 50 years later, and we made progress, for sure. We've definitely made progress. But I'm gonna predict that in your lifetime, not mine, but during yours, there will be incredibly effective ways of dealing with cancer, or maybe preventing cancer, because they're very close now to getting to where they can prevent it. But being able to treat, you look at breast cancer, the treatments for breast cancer have really improved dramatically. What are the two big types of cancer in here? Anybody?
Benign and malignant.
Say it?
Isn't it benign and malignant?
Yeah, but what I'm saying is that if we're talking about organs or parts of the body, what are the two big types of cancers? Breast cancer is one of them. Skin cancer.
Liver.
Skin cancer, we're talking about deaths.
Are you saying like the most fatal, or just
Yeah.
liver, pancreatic cancer?
Well, those would be the most fatal, but I'm talking about the number of people affected. So breast cancer for women, for men, it's what? Prostate cancer. For men, it's prostate cancer. And you cure those two, you've taken a huge chunk out of the cancer load that's imposed upon us as a society. And there've been really good progress in both of those. Okay, I gotta get to this figure. Yeah, but my prediction is that during your lifetime, you will see these effective cures come to be. The big challenges for you, and by the way, in the year that I've been alive, I'm going to say that there's the low-hanging fruit. The low-hanging fruit are the things that are the most obvious to take care of. But now all the low-hanging fruit in terms of medical challenges has been picked, so to speak. And that leaves all the high-hanging fruit, which means the very complex things are very hard to solve. But they're getting there. And I'm encouraged in terms of what I see. The biggies are cancer, the immune system. If you can maintain a person's immune system so that it's a young immune system, that's going to protect a lot. It's going to protect the heart, the blood vessels, the brain. If we can effectively deal with Alzheimer's, those are the biggies. Heart failure, huge. Okay, let me put this diagram up there. And I'll tell you, for me personally, I've lived a very blessed life, because every day I'm in the world of healthcare and I'm talking to somebody about some medical breakthrough or some type of experiment that's designed to create a breakthrough. And for somebody who loves science, it's a great life to have. So here's this diagram, and by the way, you can go out onto Canvas and you can find the diagram out there. Are you in the back there, are you guys able to see, like, here, the word triglyceride? Yeah, no? No? Okay. All right. Appreciate the help there on that, so let's try the following. How about now? Is this a little bit better?
Yes.
Yeah? Okay, so I want to talk about the breakdown of sugars first. And so with that, we're going to start up at the mouth. And as it turns out, and this is fair game for the test number 1, in your mouth you've got saliva. And within the saliva, there's a type of enzyme, a category of enzymes known as amylases. Amylases break down carbohydrates into disaccharides, monosaccharides. So up here you can see that the nutrients combine with salivary amylases, so the breakdown of sugars begin in the mouth itself. And then the food comes down into the stomach, and then from the stomach, the food enters into the small intestine. We've got the duodenum. Associated with the small intestine is the pancreas. The pancreas is this incredibly important organ. Probably, if I were to ask most of you in here, you'd tell me that when you think of the pancreas, you're thinking about insulin. But the pancreas is playing a central role in the digestion of our food molecules. So as these sugars come on down into the duodenum, the pancreas secretes what are known as pancreatic amylases. So we've got salivary amylases and we've got pancreatic amylases. And these amylases are going to break down our carbohydrates into disaccharides. And as our food continues to transit through the digestive system and moves into the jejunum, these disaccharides are acted on by a type of enzyme known as disaccharidases. So just take the word disaccharide and add on the suffix ase. Those are the enzymes that are responsible for acting on the disaccharide, and they break down the disaccharide into monosaccharides. And what's important to realize is that these disaccharides are broken down to monosaccharides, and then the monosaccharides are absorbed. What do we mean by absorbing? What we mean by that is we're putting it into the blood. And now we can use the blood to carry these food molecules all throughout the body to the cells that are in need of energy. So when you see these arrows exiting out of the gut, understand that what that means is that these molecules are being put into the circulatory system. And we're now going to use the circulatory system as a means of trying to deliver these energy molecules to the cells themselves. And that's what I need you to know with regards to sugars. All right. As part of our food, we consume proteins. And proteins are fairly large molecules. Pretty large. And within the stomach, there's an enzyme known as pepsin, P-E-P-S-I-N. And that begins the breakdown of proteins into what we'll call peptides. The food then transits down to the duodenum. And once again, we turn our focus onto the pancreas. And the pancreas secretes a category of enzymes known as proteases. And that's a good word because hopefully within that word you can see the word protein, or imagine the word protein. So there are these pancreatic proteases that break down the proteins for the peptides into amino acids. By the way, I need you to know the enzymes that are responsible for the breakdown of these food molecules, where in the digestive system that's occurring. So you can see that in this diagram is showing our protein being broken down into these individual amino acids through the action of proteases, and then at the level of the duodenum we're absorbing these amino acids into the circulatory system. Those amino acids are going to be transported to the cells of the body. What's the word that I've used to describe the machinery of cells? Nobody? Anybody? Proteins. Proteins are the machinery of cells. So what are we going to do with these amino acids? We're going to absorb them in the blood, and then we're going to take those amino acids and transport them to cells. Those cells are going to take up amino acids, and they're going to use those amino acids to make proteins within the cell itself. And also, if needed, they're going to use those amino acids to help make ATP. But I need you to understand that we've got a protein, we're breaking down the protein into its individual building blocks, amino acids, we're transporting those to the cells, and what the cell's doing with it? They're making proteins. And then, last but not least, we have our fats. And so, as the food comes in, here in this diagram, it's showing the fats, the lipids in the form of triglycerides. There are other fats, but triglycerides are really what we're focused on. And as I mentioned to you, triglycerides are a type of acylglycerol. We've got monoacylglycerols, we've got diacylglycerols, we've got triacylglycerols. Triacylglycerol is simply a molecule of glycerol with 3 fatty acids attached to it. That's represented here. And the pancreas secretes a class of enzymes known as lipases. So if you take the word lipid, get rid of the ID and put an ASC on it, we've got this class of enzymes that breaks down these fats. And you can see here in this little cartoon that the triglyceride is being broken down into monoglycerides or monoacylglycerols plus individual fatty acids. These then are transported out over into the jejunum, and the fatty acids are absorbed into the circulatory system, as are the monoacylglycerols. And that's really it. That's the basic information that helps build this part of our diagram over here. If it hasn't been obvious to you, the very first night we started out with a simple diagram, and all I've been doing is building this diagram so it's getting more and more complete moving forward. Oh, should have done that. There's one last part to that file. So there's this diagram that you need to know. And then at the end of this, I need you to know this part. So we can talk about two types of adipose tissue. There's a white adipose tissue that most frequently we're talking about. So if we're talking about subcutaneous fat in all of us, any one of us, we're really talking about white adipose tissue. But there's another type of adipose tissue that's referred to as brown adipose tissue. And as it turns out, for each and every one of us, when we were first born, there was a fair amount of brown fat in our bodies. And that brown fat plays a very, very interesting physiological role. So here, you'll notice I've got that brown fat is abundant in newborns. If you're interested in other animals besides yourself, for instance, hibernating mammals, you'll find that they have a lot of brown fat in the adult state. For us, as we undergo development, the brown fat pool decreases tremendously. What's the primary function of brown fat? And one of the functions is to generate heat. Let me just take a look at the next slide real quick before I go any further. Yeah, so here it says newborn 5% of body weight is in the form of brown adipose tissue, decreases as a function of age, and here it's just showing you where it's localized. I'm not going to test you on that. I do need you to know that there are two different types of brown fat, of adipose tissue, and one type is brown adipose tissue. And what's it involved with? What's one of the key functions of brown adipose tissue? Thermogenesis, the ability to produce heat. So just bear with me for about another 10 minutes or so, and then we'll call it quits here. So here's our mitochondria. We've got the outer membrane. We've got the inner membrane. We've got cytochrome B. We've got cytochrome C. We've got cytochrome A3. And then we've got our proton pump, our hydrogen ATPase pump. And make sure you understand that we're building this huge proton gradient so we can drive this pump backwards. And by making this pump run backwards, we end up making ATP. So we're using the electron transport chain to build this huge proton gradient. And I'm just representing the presence of this by showing all of these protons in the intermembrane space and fewer within the matrix. Now, one of the things that I did not tell you is that the breakdown of any of these molecules, carbohydrate, lipids, or fats, to make ATP results in the release of heat also. So while we're making ATP, the result of some of these biochemical reactions is the production of heat. So here's what's cool about brown adipose tissue. So all of you know that when you get really cold, you start to shiver. And when you start to shiver, what that really means is that your muscles are being almost involuntarily active to contract, to create that shivering. And by doing that, since they're contracting and relaxing, they're burning up what? Come on, they're burning up what? They're burning up ATP, and as you burn up that ATP, what's released? Energy in the form of heat. That's why you shiver, is to produce heat, to protect the core temperature. Now, this is a true story. True story. My daughter, she's pregnant. It's time to deliver, except that it's during COVID. And she's going to deliver her son, Tyler, 3 weeks early. So he's a little bit premature. And so you've got this little infant, this little neonate, and it's gone from mommy's protective environment that's producing all the heat to maintain the little baby's core temperature to now all of a sudden it's out in the air all by itself. And the heat that's producing is being lost out into the environment. And it turns out that for premature children, for premature babies, their ability to shiver is not developed yet. So they don't have the ability to shiver and produce heat like we do. And their development of the brown adipose tissue is not as well developed. Why is that important? Because brown adipose tissue has something really cool in it. And that is, it has what are known as uncoupling proteins. I'm going to just show one right here. I'll show a couple actually. We're going to call these uncoupling proteins. Otherwise known as UCPs. And here's what they do. Here's what they allow. So remember, as we're taking our glucose molecule and we're trying to extract energy from it, we're producing heat. These protons, when the uncoupling proteins are not present, our protons are going and making this proton pump run backwards, and we're getting ATP as a result. However, in brown adipose tissue, they have this unique feature they have the presence of so-called uncoupling proteins. What are the uncoupling proteins doing? Listen carefully. They're uncoupling the proton gradient from moving the proton pump backwards. They're uncoupling the production of the proton gradient from the production of ATP. You have to make X amount of ATP, but if I'm bypassing that pump and allowing some of the protons to leak back into the matrix through these uncoupling proteins, will we be making as much ATP? No, but we have to make X amount of ATP. So that means we have to have a greater flux of electrons through here. We have to burn up more glucose so that we can produce the amount of ATP we need. But we've got this short circuit now that's put in place. And this short circuit causes the cells in brown adipose tissue to make more what? To make more heat. To make more heat because we've short-circuited the production of ATP. So we have to have a greater flux of glucose being broken down with heat being liberated from it. So there's what's called non-shivering thermogenesis. Oop, that was good. And this is what brown adipose tissue does. It produces heat, but it's not due to shivering, it's due to the presence of these uncoupling proteins. So coming back to the story of my daughter, she's got her son, she gives birth to him, but he's a little premature. His brown adipose tissue is not as well developed as it would be in a full-term child. And so his ability to produce heat was reduced. Now you can imagine, this goes for both fathers and mothers. If you're a parent in here, you'll know, hopefully, or you'll feel the same way that I do. And that is, if something serious happened to your child and you had a celestial maker show up and sit there and go, your life or theirs, you know what? That's a nanosecond decision. If it's related to my sister, I'm going to tell the celestial maker, can I have a couple weeks or months or years to think about it? That's how important kids are to you, because kids are like the heartstrings of your body. So my daughter, she sits there and sees her little son. She's expecting she's going to get to cuddle him and nurse him, and now they come along and go, hey, we're going to take him down to neonatal ICU, and we're going to put him in an incubator and keep him nice and warm. But you can imagine, as a parent, you've got all these expectations of what birth is going to be like, and then you get to that moment where your child is going to have to go down and you hear neonatal intensive care unit, and you're going, holy shit, what's wrong with my little baby? You know, and you get very emotional. But the cool thing is that this is all physiology, it's all biology, it's simple to help that child nurse them along for a week or so. And then they get the brown adipose tissue, they've got their full ability to undergo non-shivering thermogenesis and protect their own body temperature. It's really cool physiology. So this thing of brown adipose tissue is important because of these uncoupling proteins. It uncouples the use of glucose and the production of our proton gradient from producing as much ATP. So we still got to produce ATP. That means that we've got to have a greater flux of electrons through here. We've got to protect this proton gradient, but to do that, we're going to actually produce more heat than normal. And that's how little babies protect their core temperature. It's really an amazing story. Okay, I've talked long enough. I'll see you guys upstairs. We'll stay in about 15 minutes. Take a breather. If you got questions,